Bismillah wa salatu wa salam ala Rasulillah. Lecture 3 will be covering Chapter 4, the human genetic diversity, and in details we'll talk about the genomic variation. We have established earlier that the human genetic is diverse, and this diversity stems from what we call variation that occurs at either at the level of a single nucleotide or stretches of the chromosomal segments. The variation can be divided into a common or rare variation. It can be entirely benign, but can also play a role in disease or disease susceptibility. So in this chapter, we will focus on the definition and the nature of variation and how it is created through mutation. So if you have two unrelated humans and you have done nuclear DNA sequence, you will be surprised that almost 99% of their DNA sequence will be essentially identical, and only a small fraction will be different, and that small fraction is responsible for the huge genetic variability between individuals, and that's a result of what we call variation. So the variations can occur at any stage of protein synthesis, from the gene organization, nucleotide sequence, DNA copy number, or protein structure and amount itself. And that what we will be discussing in this chapter of how these variations differ and how they are created through this process, and how eventually they will be divided into common variations that are not related to any diseases or rare variation associated to some diseases. Before we move forward, we will set some definitions. The locus is a specific position on a chromosome. So هي قطعة من الكروموسوم إلى عنوان معين. And it can range in size from a single gene to multiple genes to a single nucleotide. So if we look at any locus, it will have a DNA sequence. The different versions of DNA sequence at a specific locus, we call them alleles. So for example, if we look at a specific locus and the DNA sequence, for example, was A, B, C, and there's another version, CBA, for example, we call each of them an allele. And that can be divided into wild type or common allele. So for example, if the ABC sequence is present frequently, and by frequently I mean it's in more than 1% of the population, this version is called a wild type or a common allele. And if a mutation happens at that locus and that resulted in a different DNA sequence, for example, CBA, and by mutation I refer to changes in the DNA sequence, but that doesn't imply any specific function or fitness or on that location or on that sequence. We call the sequence a variant allele. And for example, if a CBA, for example, was a sequence that is not common and it's present in less than 1% of the population, we call it a variant allele. And the frequency of different variants can vary widely in different population. So sometimes we refer, like there is a word called polymorphism, when a locus have two or more relatively common alleles in a population. Or sometimes the variant alleles, they will be very rare, that will be present in only a single family, and in that we will refer to sometimes as a private allele. How would you define a variant which is different from the common or different from the normal? And that raises the need for a gold standard or a reference sequence or genome, which is defined as the most common sequence or arrangement in a population at any one position in the genome. And this reference sequence is subject to constant evaluation on change. However, there are several international collaborations that made their data access to public, defining the reference sequence or the gold standard sequence. As examples of these collaborations are the Human Genome Project, the 1000 Genome Project, or the Human Gene Mutation Database. So the variants are sometimes classified by the size of the altered DNA sequence, and that can happen at subclassified into three different levels. It can happen at the level of chromosome number. So the chromosomes will be intact, but the changes will be in the number of chromosomes in the cell. Or it can happen to only a portion of a chromosome, what we call a change in the subchromosomal segment or structural rearrangement involving parts or more of one chromosome or more than one chromosome. And we refer to these as regional variation or copy number variation, CNVs. Or that alteration can happen in the sequence of DNA. And that will include, for example, substitution, deletion, or insertion of either one or a single unit or a triplets of nucleotide, and we will discuss these details. The common variants are important. As we said before, we set the definition of a common variant that it is a frequent variant in a population that happens in a frequency in more than 1%. But what are the uses of common variants? The study of common variants become very important recently because we are using them in daily practice. We use them in research tools to map the genes in specific chromosomes or regions. We use them in prenatal diagnosis, detecting genetic diseases during pregnancy. Also in blood banking and tissue typing, especially now becoming more interesting in organ transplantation or bone marrow transplantation. And also it has some forensic application to identify the paternity or in the crime scenes. And we have been using them, like especially in my specialty now in precision medicine. So we tailor the medical care based on individual or genetic variants in specific tumors. And the common example now is the lung cancer, that using the common variants to tailor the treatment. So in the next few slides, we will discuss the common variation in human genome, some inherited common variations that will include the single nucleotide variant, insertion, deletion, or indels, or copy number variant, and inversions. So single nucleotide variant, as the name implies, it happens at the level of a single nucleotide. This is an example where the reference sequence in allele1, the T is changed to C at the position number 8. The common SNVs happen in higher population frequency, more than 1% of the population, but can happen at anywhere in the genome, but and many are found in non-coding parts of the genome, such as introns, or sequences that distance from the protein coding genes. So most of the SNVs, like I would say more than 50%, they don't alter the predicted amino acid sequence and or the encoded protein, and we call them synonymous SNVs. And those that alter the amino acid sequence, we call them non-synonymous SNVs. And you can predict that if that change is in the SNV, in the single nucleotide, change around the stop codon or a splice site, it will cause it will have more consequence of affecting the function of the protein or the function of the DNA sequencing. So other common variant is the insertion deletion variants. These result from insertion or deletion, what we call indels of segment that ranges from a single base to up to 1 kilobase. So over 1 million indels have been described in the human genome, and approximately half of all indels are referred to as simple indels because they have only two alleles, that is the presence or absence of inserted or deleted segments. This is an example of insertion deletion things. So for example, in indel A here, we notice an insertion of a G between the positions 11 and 12 to the reference allele. So G here is added between 11 and 12. This is another example of deletion variants. The indel B, the LL2, has two base proteins deletions as positions 5 and 6. So the T here has been deleted from that position. So the indels happen sometimes at a larger scale, larger than a single nucleotide, and in something we call microsatellite variants. And these microsatellite variants are larger indels which are usually multi-allelic due to variable numbers of segments of DNA in a tandem. And that's why we call them sometimes called the variable number of tandem repeats. And these microsatellites are highly vulnerable to mutations. They consist of DNA cassettes composed of units of several nucleotides. This is an example of one of them. For example, the microsatellite locus has 3 alleles and with 4, 6, and 5 copies of CAA trinucleotide repeats. And the number of repeats units determine the different alleles, and sometimes we refer to these as short tandem repeats. They have high importance in clinical applications, especially in genetic mapping and DNA fingerprinting. Another example of larger variants is the mobile element insertion variants. You have to know that the human genome consists of dispersed families of repetitive elements. Most of them are stationary, they don't move, but a small proportion um, of the RNA undergoes what we call a retrotransposition. So the RNA will go into a process of reverse transcription into DNA, and that DNA sequence will be inserted into another site of the genome. The two most common mobile element families are the ALU family and the line family, the long interspersed nuclear elements families repeats. Each have two alleles, the allele one without the inserted mobile element and the other one with the inserted mobile element. And you have to know that most of these mobile elements happen in the non-genic region. A small proportion of them are found within the gene. Another type of larger variants are the copy number variant, which essentially it's an indel or microsatellite, but happens at a larger scale and a larger segment of the genome, and that will be up to 3 million base pair. The large CNVs or the copy number variant sometimes happens in regions of genome characterized by repeated blocks of homologous sequence called SIGDUPs or segmental duplications. The smaller CNVs may have two alleles. For example, this is an example shown here. There was allele1 contains a single copy, where allele2 contains 3 copies of chromosomal segment containing the F and G genes. But there are other examples or other alleles that can be identified where the allele3, for example, will have only 2 copies of that or 2 repeats of that FG segments. And you can relate that the CNVs will cause more genome diversity than SNVs because it involves a larger amount of DNA. And the last type of larger variants that we'll talk about is the invergent variants. Invergent variants, basically, there will be region in the genome, and that will be either few base pairs or several megabases are found in either two directions. This is an example here in allele1 CDE in that direction. In allele2 it's inverted to EDC in the other direction. And if there is no loss in DNA through this variation, we call it balanced variation. And but sometimes this will be associated with some deletion or duplication of DNA. In this process, it can be associated with some clinical disorders we'll discuss later. So as we said, the mutations, they are events that will alter the DNA sequence at a specific locus. And The mutations occur during fundamental processes such as DNA replication, DNA repair, recombination, and chromosome segregation during cell division, including mitosis and meiosis. We divide the mutations into inherited or somatic mutations. Inherited variations originate as de novo mutations in germ cells, while somatic mutations occur in different proportions of cells throughout the body and cannot be transmitted to the next generations unless it is a germline mutation. And to identify these mutations, what we use in the clinical settings, we use something we call DNA sequencing to identify these somatic mutations. And as I mentioned earlier, one of the most important or like the most applications of the DNA sequencing and identifying the somatic mutations is identifying somatic mutations in some tumors, such as lung tumors, for example. And currently, we are using this to tailor treatment based on detection of specific mutations in the DNA sequence. So what we will be discussing for the remainder of this lecture is the origin and frequency of different types of mutations or rare variants. One of the most common source of variation seen in humans is the chromosome alteration that happens during chromosome uh, segregation. The missegregation will affect the number of chromosomes, and this is one of the most common causes of spontaneous abortion that happens that we see in clinical life. Other levels that can happen is the structural variation at the structure of a region of a chromosome, and that result from duplication, deletion, or inversion of a segment or of a single chromosome. Or these variation can happen at the level of genes. So gene or DNA variants includes base pair substitution, insertion, or deletions can originate in either or two basic mutational mechanisms, either during the DNA replication, so introduced error during replication, or if the replication happens and error happens, there is a failure of properly repair that DNA damage, either during processing of DNA replication or and repair, or induced by physical or chemical agents called mutagens, happens later on. Subhanallah, the DNA replication is a process that is very highly accurate, and most errors rapidly corrected by a DNA repair enzymes. We call them usually DNA polymerases. This happens after what we call DNA proofreading. The process recognizes and corrects incorrect bases in the newly synthesized double helix, replacing them with a proper complementary bases. And the error in this process is about in 10 million base pairs. And you have to know that there will be additional proofreading, corrects more than 99.9% of replication errors, resulting in an overall mutation rate of approximately 1 times 10 to the power of minus 10 per cell division, which translates to fewer than one mutation per genome per cell division, which is very, very, very rare occasion. Accuracy applies also on the repair of DNA damage. We estimate about 10,000 to 1 million nucleotide are damaged per human cell per day, and that due to either spontaneous chemical processes such as depurination, demethylation, or deamination, or reaction to a chemical mutagen or exposure to ultraviolet or ionizing radiation. So these all will cause damage to the DNA. Some, but not all of the damage is prepared, and even if such damage is recognized and excised, sometimes the repair mechanism may introduce incorrect bases. So we assume that the nucleotide changes introduced by DNA damage and repair are often permanent, and one of the most common spontaneous mutation that happens is the T4C substitution. We will mention that later. You don't need to know everything on this slide, but this is just to tell you how we measure the rate of new mutations, that we measure the incidence of new cases of genetic condition that is clearly recognizable in all neonates who have particular genetic alterations. And we get an example here of achondroplasia that is measured on the locus FGFR protein. What I need to highlight here that there is an effect of the sex and the age of the mutation rates. So because that the DNA goes far more replication cycles in sperm compared to the ova, that means that there is a suggestion that the new variants will be more often paternal, coming from the father, than coming from the mother in origin. Furthermore, because of in the man at old age, he will have more rounds of replication prior or preceded the meiotic divisions. That means that the frequency of new paternal variations might expected to increase with the age of the father. So being a father, being an old father specifically, that will increase the incidence of having new variants. We will slightly expand on some types of variations. Some is related to the nucleotide substitutions, just like such as missense variants, nonsense variants, DNA processing variants, splice size variants, long range regulatory variants, and some associated with deletions or insertion, like additional deletion of small number of bases, larger gene deletions or inversions, insertion of line or ALU element, or dynamic variants. Okay. The best example of nucleotide substitution is the missense variants. So there will be a single nucleotide substitution, or what we call a point mutation in a gene sequence. Such events are called missense mutations. They will create variants because they alter the coding or the sense strand of the gene, specifically result in a different amino acid. Not all missense variants lead to like an observable change in the function of the protein, but they will result in a protein that is weak and they fail to work properly and may be unstable and rapidly degrades. One of the examples of these missense variants are the achondroplasia. So another example of nucleotide substitution variant is the nonsense variant. Similar process, it's a point mutation of a segment of the gene, but the problem here, it's the normal codon will be replaced by a termination or a stop codon, and creates what we call a nonsense variant or premature termination codon, PTC. Okay, so that will cause the stop of or termination codon causes the translation also to stop permanently. In general, the mRNAs harboring these permanent termination codons um, are targeted for rapid degradation through a cellular process called nonsense mediated mRNA decay, so NMDs. Okay, you have to know, in rare occasions, what happens is that alteration that happens, like the nonsense variants, will cause the stop codon to change. And in that case, will cause the exact reverse. So there's no stop codon anymore. So the function or the protein transcription will continue until a stop codon comes in the natural sequence of the gene. So Another example of nucleotide substitutions are a variant affecting RNA transcription, processing, and translation. And the basic concept is a pure point mutation that will change the codon, but that codon will include sites, include transcription factor binding or 5' capping or polyadenylation or splicing. Um, so it will become a non-coding RNA or a mature mRNA, and that will cause the affect the mRNA stability, translational efficiency, and reduce the amount of protein produced, potentially contributing to some diseases as well. The other family of variants are the deletion, insertion, rearrangement that may involve few nucleotides detected by direct sequencing or part of the genome, or may involve a substantial segment of the gene. What we call them like structural variants. Two things just to mention here. Sometimes we have what we call a frame shift variants, where the insertion and deletion of number of base pairs is not divisible by 3. ليس من مضاعفات الثلاث. What happens here, if you lose 3 base pairs, that will affect one amino acid, and it will not affect the entire sequence, but it will entire one amino acid. But if you lose 1, 2, 4, that will cause a frame shift, and that frame shift will result in the transcript of degraded or truncated protein products. So that's why insertion or deletion of something that is not divisible by 3 is more dangerous than things that can be divided by 3, which also can affect the amino acid alteration, but it will affect one amino acid rather than change the whole sequence. The other things are the insertion mutations that we mentioned earlier in the common variants that can happen here as well. So insertion mutation that involves insertion of a mobile element like line 1, so L1, which can cause genetic diversity, but also can cause some diseases through the insertion of mutagens. And that an example is the gene causes hemophilia A that is associated to the line 1, or some of the mutations that happens in colon cancers. We show an example of larger scale genetic changes, which include duplications, deletions, and inversions of chromosome segments result in homologous recombination. This is an example of factor 8 gene, the upstream mispairing and recombination of a segment of a gene that will cause the hemophilia A mutation and inverted segment within a gene. هذا مش مطلوب. This is not for memorization. مش لازم تحفظه. I just want you to imagine the amount of variation that happens in an individual genome, and that's subhanallah, this is the reason why we are diverse, and we have diversity amongst individuals, because each individual have a different or yeah, at the genome level will have lots of variation that happens. For example, you will have about 3.5 million SNVs compared to the reference genome, and you have to know that what we don't know is even far beyond that we do know. With each sequence of a gene, we discover new mutations and new variations that we haven't discussed before, and we keep on documenting. And that's why I say that these large genome libraries are libraries that are dynamic and undergo ongoing changes. So in the clinical work, sequencing is used nowadays, especially in cancer patients. We use two types of sequencing, either studies of the entire genome, like whole genome sequencing, or we study a subset comprising all non-coding exons. For example, in lung cancer, we do a limited panel that will include specific mutation looking for specific sites, specific loci on specific chromosomes, such as EGFR, KRAS mutation, BRAF mutation, and etc. In melanoma, for example, we test for a BRAF mutation. So and that will look either in germline or a somatic testing. For example, Lynch syndrome, we do some sequencing for Lynch syndrome, for example. But because, yeah, now everything is becoming commercial, we have non medical or commercial sequencing of genome, which what you hear, like people will do genetic testing to know their ancestries, to do their origin genes, as well as some personal and health traits. We will wrap this lecture up with a few points. The impact of variation. So rare variants known to have clear clinical consequences. The common variant don't. However, sometimes the common variants can be clinically significant. Um, Individuals are likely to carry two distinct alleles for many proteins coding loci, leading to what we call polymorphism. And even if the gene product remains unchanged, sometimes the expression level can vary among individuals due to genetic and epigenetic variation. And that leads us to the point that each person has a unique genetically determined biochemical makeup, leading that each person has individual response to environment, dietary, and pharmacological influence. So if you go down and do a whole genome sequence and you identified variants in your chromosomes, in your genes, these variants are now classified into 5-level scale. So they are either pathogenic variants, they are likely pathogenic variants, they are of uncertain significance, they are likely benign, or definitely benign variants. And now in each whole sequencing, or even the targeted sequencing that we do in our labs, we put a comment to identify the category of that variant, or the significance. We know that, for example, I keep on putting this example in lung. EGFR is of a clinical significance because in lung tumors, because it can affect the treatment, it can direct the treatment. In the other hand, there are other mutations like TP53 mutations. We don't know the clinical significance. We know that it is present in tumor, but we don't have a targeted therapy to it. So that's now it will be put in an uncertain significance. So this is how we scale those variants that we see or we recognize during the sequencing. And to sub-classify into those mentioned categories, we depend on the assessment criteria. Again, you don't need to know the exact details of single one, but we depend on the population frequency, the in silico assessment, in the functional data, the segregation data, the de novo variant or variant characterization or prior occurrence to determine if that it is pathogenic, significant or non-significant. But no single criterion is definite, and they must be considered collectively to determine the pathogenicity of these variants. And this is the end of my lecture. Please direct your questions out to this email or the WhatsApp group that we will be creating.